So, it's not, don't worry about me. I have lots of spaces left. Anyway, so to begin with, we're going to take a look at how information is encoded in DNA. And so I just want to walk you back really quickly to make sure we're all on the same page, which is the reason we started talking about the central dogma, moving from DNA to RNA to proteins, was because this is how information is encoded in DNA. So we talked about DNA replication so that we can make complete copies of DNA for cell division. Now we're talking about what the cell uses DNA for when it is not dividing. Okay? So we have a set of terminology that we use, and because it's science, we've given the same strand of DNA 3 different names. You have to be cognizant of all 3. They mean the same thing. Okay? When we talk about information on a piece of DNA, we say that the information is encoded on the antisense strand, so we can call it the antisense strand. We also refer to the exact same strand as the template strand, and we also call it the non-coding strand. Okay? So, if you take a look at our piece of DNA, for example, we have a blue piece of, well, it's not on yours, I'm sorry. On yours it's not blue, red, and green. But on mine, you can see it's blue, red, and green. Sadly, we can't give you color copies. But you'll notice that I have a blue strand that's considered the antisense strand. I have a red strand that's considered the sense strand. As far as directionality is concerned, you'll notice that the sense strand moves from 5' to 3', and the antisense strand is running 3' to 5'. Okay? It makes sense. We've got anti-parallel pieces of DNA. Here's the other rule you need to know. When RNA is constructed, it is constructed by a polymerase. Instead of using DNA nucleotides, though, this polymerase uses RNA, but it's still a polymerase. So what rules does the polymerase enzyme have to follow? Because they will be the same as its DNA counterpart. All right, so it has to build the strand. It has to build the strand 5' to 3', right? Because polymerases can only add new nucleotides, whether they're RNA or DNA, to an existing 3' end. And if RNA functions, or structurally functions the same way as DNA, if I have to make the new strand of RNA in the 5' to 3' direction, what strand direction is the DNA that we're building on? It's going to be opposite, right? We have to, again, preserve anti-parallel structure. I cannot flip the direction of the messenger RNA that's being synthesized. Can't do that because the polymerase can only work 5' to 3'. That means that my template strand must always be 3' to 5'. Okay? So if we're using the sense terminology, then the sense strand, which runs 5' to 3', is not going to be used to make the RNA. Rather, we use the antisense strand. And the piece of messenger RNA that's made will be synthesized from the 5' direction to the 3' direction. We call this the sense strand then because the sense strand will run the same way as the messenger RNA. The only difference is that there will be T's in the sense strand and U's in the messenger RNA. But otherwise, sequence-wise, it's the same. Does that make sense? Okay. If we use the terminology for template or coding strand, then the template strand is 3' to 5', and its complementary strand is called the non-template, or it's also called the coding strand. Occasionally I've heard, though it's not very often, I actually have heard faculty members refer to this as non-coding. But if I'm using the non-coding or if I'm using the template terminology, then it's 3' to 5'. The other stranded DNA is referred to as the non-template, or it's also referred to as the coding strand. So the coding strand will not give me a piece of messenger RNA, and the non-template strand will not give me a piece of messenger RNA. But the template or the non-coding strand will. Same thing happens here. we use the 3' to 5' DNA template to make a 5' to 3' piece of messenger RNA. And this piece of messenger RNA, the sequence will match the non-template or the coding strand, with the exception that all of the T's will be replaced by U's. Okay? So it's the same concept, we're just giving it 3 different names. Okay, because that's science. When you read the literature, it just depends on what kind of day they're having, whether they're using sense or antisense, template or non-template, coding or non-coding strand. Okay? So look at it one more time, this time with coding and non-coding strand. So the coding strand is the 5' to 3' piece of DNA. The non-coding is the 3' to 5' piece of DNA. And that's used to make your messenger RNA, which is 5' to 3', and whose sequence will match the coding strand. Okay? Does that make sense? You're okay?
Just have a question. On that image for the, I think it's the antisense, shouldn't it be 3' to 5' for the mRNA? Do you see what I'm saying?
No, because the mRNA has to be built in the 5' to 3' direction. So the piece of messenger RNA is being made 5' to 3', and you can see the arrows indicating which way the RNA polymerase is going. I can't build a piece of messenger RNA 3' to 5' ever because it's a polymerase. But as a result of not being able to build it 3' to 5', that means that as a result of not being able to do that, that means the 5' to 3' piece of DNA can never be the template. It has to be the 3' to 5. The DNA template has to be read 3' to 5' because the messenger RNA has to be made 5' to 3'. And it has to always be anti-parallel and complementary. All right, so just like its DNA counterpart, messenger RNA synthesis has to occur 5' to 3'. So polymerases only add to the existing 3' ends. The difference between DNA polymerase and RNA polymerase is twofold. The first is that obviously RNA polymerase doesn't incorporate DNA nucleotides. Instead, now it's incorporating RNA nucleotides. The other major difference is that unlike DNA, RNA polymerase does not require a primer. So it can initiate RNA synthesis at the start of a gene. So I don't have to lay down a primer to provide a space for the RNA polymerase to attach to. Instead, the RNA polymerase begins at the initiation site for transcription. Okay? Now, we were talking about the DNA template being read 3' to 5' in order to synthesize the piece of messenger RNA. And if you think about it, then that means that if the DNA is only encoded on the 3' to 5' strand, then that would mean that only one strand of DNA is actually used to make messenger RNA. And that's not the case. Genes can be encoded on either strand. The only thing they have to preserve is directionality. So for example, if we take a look at gene 1, here's my RNA polymerase. Okay? RNA polymerase, by the way, is multifunctional. It has a helicase inside of it, so it's going to unwind the DNA, synthesize the RNA polymerase all in one go. Okay? And because it doesn't use single-stranded binding proteins, as soon as the RNA polymerase passes by the piece of DNA, the DNA is able to rebind to itself and retwist into a helix. Okay? Does that make sense? And then you'll see the growing strand of messenger RNA, okay, as the RNA polymerase synthesizes. If we take a look at gene 1, my messenger RNA is being made 5' to 3', which means that this piece of DNA is running, this is the 3' end of the piece of DNA, and this is the 5' end of the piece of DNA. Right? The other strand will also function as the template strand for another gene. All I have to do is make sure that the RNA polymerase is running in the opposite direction. So for gene 2, okay, instead of using this bottom strand as its template strand, instead the top strand is the template strand. But this time, instead of opening the fork in this direction, RNA polymerase is opening the fork in this direction. So I still preserve the ability to make a piece of messenger RNA going 5' to 3'. If this is the 5' end, then on the template, this must be its 3' end, and we head towards its 5' end, okay? So either strand can function as the template strand. It just depends on which direction the polymerase is operating in, okay? So do keep that in mind. It's not that there's like one whole strand of DNA that we never use that's total junk. Okay, we use both strands. It just depends on the directionality that the polymerase is opening in. Okay? So if we take a look at what our RNA polymerase is doing, remember, same thing we had going on with DNA. We have to maintain the anti-parallel structure. So this is the 5' end of my DNA template strand. This is the 3' end of my messenger RNA. So it maintains anti-parallel structure. Okay? You'll also notice that we maintain complementary base pairing. See that? And the only difference is, again, that instead of having a T, messenger RNA would insert a U. It inserts the U across from a DNA template A. So just keep that in mind. Remember the base pairing rule that we have to associate. So if we have a piece of DNA and it's A, what's the piece of messenger RNA going to have? Yeah, it's going to have a U, not a T, right? If the piece of DNA is T, though, what will go in for the messenger RNA? A, right? The only thing that gets replaced in the messenger RNA is the U. Okay? Does anything change with our G's and C's? If it's a G, messenger RNA puts in a C, and if it's a C on the DNA side, messenger RNA is going to go ahead and put in a G. Okay? So far, so good? All right, so the trick, remember we said, is that RNA polymerase doesn't need a primer, because when we think about genes, they're found in something called a transcription unit. Okay, so let's take a look at the transcription unit. So you think about this being a block of DNA that encodes your gene. All right, you'll notice I have two strands of DNA. I have a 5' to 3' strand and a 3' to 5'. Okay, we're identifying the template and the non-template for you now. Just so you know, on the exam, you will have to identify which is the template and the non-template strand, okay? And we're showing you some very special regions. First thing let me identify for you is that we use a set of terms called upstream and downstream. Upstream means before, downstream means after. So we always say, oh, this is upstream of the promoter. That means it comes before the promoter. Or we say this is downstream of the promoter. That means it comes after the promoter, okay? You can move the site that you're talking about, and that means that it changes what's upstream versus downstream. So for example, if I choose to move the point here, right? I'm going to pretend that's my arbitrary point. Now everything that is upstream from that arbitrary point is here, right, before it. And everything that is downstream from that arbitrary point is here and behind it. Okay? So we just use upstream, downstream so that we know where we're moving along a piece of DNA, right? Because we can't use up and down or left or right, correct? Yeah, because up and down and left and right don't exist. So we use upstream and we use downstream, okay? We then have a block of DNA called the promoter. Do you remember what a promoter is? What kind of gene is it?
Continuous.
So remember we had three types of genes? We talked about three types of genes. We had structural, regulatory. Yes, promoters are regulatory genes. And they are flanked by the other regulatory gene, which is the terminator.
Yes.
Okay? So these genes contain information, but they don't actually give us a protein product. Okay? For a promoter, the information that it contains is the start site for transcription. So to the promoter, RNA polymerase is capable of binding and starting transcription. So the promoter marks the part or the site where transcription starts. It allows the polymerase to bind, start unwinding the DNA, incorporating the new mRNA molecule. You'll notice that although RNA polymerase binds to the promoter, it does not transcribe. The transcription site, the start site, is downstream of the promoter. If you read your textbook, you might hear it referred to as +1. The promoter is 0. The transcription start site is +1 because we don't start transcribing until we get to the start site. So although RNA polymerase binds here, it will not make messenger RNA. So there's no mRNA made for the promoter region. Okay? If you take a look at my piece of RNA, you'll notice that transcription doesn't start until +1 at the transcription start site. Okay? Once RNA polymerase hops on, it'll transcribe the entire unit. By the way, which strand will it transcribe? The one on the top or the one on the bottom?
It doesn't transcribe 5 to 3.
Here's my promoter. It's got to start here, so which strand does it have to use?
Template strand?
It's got to use the 3' to 5' strand. That's my template strand, because here's my promoter.
Yeah.
Okay? The transcription will occur all along the RNA coding region. This is what gives you your structural gene, because this is where the information actually is for the protein. And then it stops. When we hit the transcription termination site, the RNA polymerase will be removed, transcription will halt. But you'll notice that like the promoter region, there's no usable messenger RNA signal made from the transcription termination site. Okay? There's a piece of messenger RNA there, but we're going to get rid of it in just a second. Okay? The only part that codes for a physical protein is actually in the RNA coding region. Okay? So this is my transcription unit. I've made a piece of messenger RNA. You'll notice that it's 5' to 3'. Okay? And I've made it from the 3' to 5' piece of DNA. Okay? The promoter is the boundary. It's the start site. The terminator is the other end. So everything in between the promoter and the terminator gets transcribed. Okay? Questions? Now, this in part is the reason why we have to deal with temporal separation. Okay? This is called messenger RNA processing. So even though I've made a piece of messenger RNA, that piece of messenger RNA cannot be used immediately for translation. Okay? In a eukaryotic cell, there's 3 major steps for messenger RNA processing. So all of these things must occur before translation occurs. So it happens fairly simultaneously, but we'll list them off as if they happen one at a time. The first thing that happens is exons have to be spliced together. So the coding region of the gene is made of 2 things. It's made of introns and it's made of exons. We only need the exons. So we splice the exons together and we remove the introns. It's a bit like if we had a textbook and you're reading along the textbook and you're reading about protein synthesis, and then all of a sudden there's like 6 pages about ecology and diversity. So you're reading through these 6 pages and you're like, this has nothing to do with protein synthesis. You flip through the 6 pages and then all of a sudden we pick up again with protein synthesis. And the book carries on like nothing happened. And then 4 pages later, you get to a handful of pages that are about the human heart. You're like, what's going on? You flip through 3 pages on the human heart, and the book picks up again with protein synthesis like nothing ever happened. If you want to read the story of protein synthesis from beginning to end without any of those intervening sections, what do you got to do? What are you going to do?
Take each section that has.
Yeah, you want to take out the part on ecology and diversity and take out the part on the heart, right? Don't rip pages out of books, though. That's not nice. But that's what you'd have to do, right, to get yourself a nice story. So the body has to do the same thing. We have to remove the introns because they have nonsense information in them. If you try to go ahead and translate with the introns, you will not get the protein that you want because those sequences should not be present. So the exons have to be spliced together and the introns have to be removed. Now, to protect the 5' end of the piece of messenger RNA, we put on a methylguanosine cap. That goes on the 5' end only. And then to the 3' end, we add a poly A tail. What does poly mean? Many. So the tail is just many adenines added to the 3' end of the piece of messenger RNA. Okay? Only once you have completed all of your mRNA processing is that piece of messenger RNA ready to be translated. Once all 3 of these things have been completed, we now start to refer to the messenger RNA as mature. Mature messenger RNA that has undergone all 3 editing processes is ready to be translated. And then the spatial separation part comes because once it's ready to go, it has to leave the nucleus and head into the cytoplasm. So keep in mind that all of this editing is happening where? All of the editing is happening in the nucleus because it's part of transcription, and all of transcription takes place in the nucleus. Okay. Here's my transcription block again. I've made the piece of messenger RNA, and you'll notice we've gone 5' to 3'. Okay. This is the promoter where messenger RNA binds to, and this is the TSS, that's our +1, that's the transcription start site. So you'll notice that I don't start making messenger RNA until after I've reached the TSS, right? Because although the promoter has information for where to start, it will not be transcribed. Okay? When transcription occurs for a piece of messenger RNA, you'll notice we give you everything, right? We give you the exon, we give you the introns, we give you all of the pieces. Okay? But when we get down here to the piece of messenger RNA, remember one of the processing units is that I don't actually need the introns. These have to be removed. They're removed through a process called lariat formation. So you'll actually take these 2 exons and you'll pinch them together. And when you pinch them together, it pushes the exon out. Okay? And then you'll take this exon and you'll push them together. And when you push them together, that causes the intron to be popped out. We refer to this process as splicing. And so now when you take a look at your piece of messenger RNA, it's only made of exons, right? And the introns are removed. Okay? We then cap the 5' end, and we put a poly A tail on the 3' end, and now we consider this mature messenger RNA. Not yet. Okay? I'm going to show you one more thing. We're actually not going to use all of the messenger RNA that we've made. So as we get ready to head into translation, which is the next phase, we will only translate what is in between the start codon and the stop codon. Okay, so I know you don't know what a codon is, but just bear with me for a second.
Yes, we do.
Oh, okay. Okay, then. Wednesday, kids, you would also know what a codon is, too, except the computer program doesn't work anymore. Surprise! Anyway, everything that comes before the start codon is referred to as the 5' UTR. UTR stands for untranslated region. We also occasionally refer to these as leader sequences. They come before the translation start. Okay? Downstream, so that means after, downstream of the stop codon, we then have a 3' UTR. What does UTR stand for? Untranslated region. So this piece of messenger RNA will not be translated into protein either. We occasionally refer to the 3' UTR sequence as the trailer.
So is that just taken out? Not using it?
Nope. It's going to head, the leader and the trailer is all part of the mature messenger RNA. It's just that when this meets the ribosome, neither the leader nor the trailer will be translated. So only the regions in between the start codon and the stop codon will be made into protein. Okay? But the leaders and the trailers are still in the messenger RNA. They're just not used. They're there to protect the piece of, well, I should say they're there to protect the coding region that actually gives you the protein, because the minute the messenger RNA hits the cytoplasm, there are millions of mRNases in the cytoplasm that are breaking down the messenger RNAs. It's part of how we regulate how much protein is made. But while the messenger RNA is in the process of binding to a ribosome, we have to make sure that we protect the information. Because if a piece of RNA is destroyed before we have a chance to translate it, what happens to the protein? It doesn't get formed correctly, or it may never get made at all. So all of this junk, if you will, that never gets used is there to protect the actual region that codes for the protein. So if you get a RNase that comes in and starts degrading the RNA, it has to get through all of the 5' untranslated region, or all of the 3' untranslated region, before it actually starts degrading the signal that gives you a protein. Okay? Have you ever spilled coffee on something? I mean, maybe it's not coffee. For me, it's always coffee, but I mean, have you ever spilled? If you notice the papers that are in between usually don't get wet because all the other things protect them. But you usually have to throw away the first couple of pages because they're soaked in coffee or they're disgusting or kind of weird. Same thing happens here with piece messenger RNA. Okay? We're going to protect it from those cytoplasmic RNases by having it capped and tailed, and also having the leader and the trailer.
What does CDS?
Don't worry about it. That's a set of terminology you probably don't want to get into.
Now, when the trailer and caps are broken off, is the poly and cap are stuck to the codon, or they break off to the trailer?
So when the ribosome comes in, it's only gonna, so the ribosome can pull the entire, so let's say none of this gets degraded when it hits the cytoplasm, the ribosome's gonna pull this in, but it won't start incorporating amino acids till it hits the start codon. Once it hits the start codon, then it starts to put in amino acids. Once it hits the stop codon, it stops putting in amino acids. And then this messenger RNA is left in the cytoplasm where it will then be fully degraded by RNases. So it's not about it like falling off or not falling off. It's just not used by the ribosome. But once the stop codon is reached, the ribosome breaks apart and the messenger RNA is left in the cytoplasm where it's attacked by the RNases and broken down. If you didn't break down the messenger RNAs, what would happen?
Protein is made, right?
Well, you make a protein, and then what happens when the next ribosome comes along? It would make another protein, and then another protein, and then before you know it, you would have too much protein, right? So this is a regulatory mechanism. If we break them down as quickly as they show up, that means basically ribosomes have one chance to translate this piece of messenger RNA before they're degraded and removed. Okay? So it's how we regulate protein transcription. So if a cell needs more protein, what does it have to make more of? It has to make more messenger RNA. And in fact, in molecular biology, we measure the amount of messenger RNA that you're making as a measure of the amount of protein that you're producing. Because it's almost, in a eukaryotic cell, it's almost 1-to-1.
Where does splicing fit into the central dogma? Is that the same thing as transcription and translation?
Well, splicing is part of transcription.
Okay.
because all of the mRNA editing is part of transcription, because we make this piece of messenger RNA, but all the things that are in the DNA aren't necessarily used to make the protein. So we have to remove all the things that aren't essential for the protein, right? Rip out all those random pages, put all that information together, then we cap it, then we tail it, and then once it's mature, that's when transcription ends.
You said intron is filled with nonsense.
Mhm.
Nonsense like what?
Like sequences that are not used to make the protein. If you leave the introns in and you translate the introns and the exons, you will not get the protein that you want. It'll be all wrong. Now, what's the point? What's the point then of having so much junk? Because all of your genes, every single gene that you have, approximately anywhere between 30 and 70% of the gene signal is introns. That's junk. What's the point?
Fillers?
Part of evolution.
What's the point of fillers? Protecting me from what?
From degrading? Well, not degrading.
Well, DNA is not going to be degraded unless something very, very wrong is happening in your cells.
Makeshift, like when there's mutations and stuff.
Yes. Introns protect you from mutations. Okay? All right, friends. An average gene's couple million bases. Okay? We've addressed the idea that all I need to do is change one base to go from being perfectly normal human being to having sickle cell anemia, correct? It's one base in your DNA. Okay? Because 70% of those millions of bases don't actually code for anything, what happens if a mutation happens in those 70% that doesn't code for anything at all? Absolutely nothing at all happens. So there are only very small pieces of your DNA that are actually responsible for making the proteins, and by having random sequences of junk that aren't useful anywhere else, it protects you against mutations. Humans have one of the lowest mutation rates of any organism on the planet because we protect the functional coding sequences with so much junk. You are padded with nonsense, okay? But the nonsense protects you from all the mutations that are happening, okay? Now, what happens when the mutation happens in the little piece that you actually need for coding? Yeah, then it can be detrimental to the organism. Go ahead.
So how come, like, since other cells can mutate, like, overnight and everything like that, like, disease and everything, then they mutate, then how come sometimes they do affect us? How do they affect us?
Well, so if the mutation happens in one of these small coding regions that actually gives rise to the piece of protein that we need, then you're not going to get the right protein. And when you don't get the right protein, that's when you can start to see problems.
From that mutated thing?
Mm-hmm.
And that's how it gets in?
Yeah, so your body actually makes the mistake usually. Yeah. So we're going to talk about this, but there's two kinds of mutations. There's the kind of mutation that's passed to you through genetics, but then there's the kind of mutation that you make yourself. You guys all know what cancer is? Cancer almost universally, except for a handful of types of cancer, cancers almost universally are mistakes that your cells make that lead to the disease. There are not that many inheritable cancers out there. So it's not like you get a bad gene passed to you. Instead, you had a good copy of DNA, and then your body made a bunch of mistakes, and you landed with a copy that left you vulnerable and gave you cancer.
Is that why you get tumors?
Mm-hmm. Yep. Mm-hmm. So the good news is, though, since there's so many proteins and your cells are replicating and you're making so much DNA, you haven't made that many mistakes, obviously, since you're all sitting here with me today. So that's actually kind of an impressive feat. Right? I'm usually feeling kind of generous and in a good mood when I write your exams. So 9 times out of 10, if you've been really nice, your piece of template DNA will already be running in the 3' to 5' direction. So you don't have to do anything with it. If you stop trying in class, I'm going to put the piece of DNA in the 5' to 3' position. You'll need to flip it. No, I'll go ahead and run it in the 3' to 5'. So
Where's the love? Too much love, huh?
etc. Okay, functionally for you guys, what do we have to do next? We have to make a piece of messenger RNA, right? So you're going to make a piece of messenger RNA. Now you know your piece of messenger RNA has to be anti-parallel. So if I've given you the 3' piece of the DNA, that means your messenger RNA starts with its 5' end. Okay? So we know it's anti-parallel. Just took care of that for you. It also has to be complementary. Okay, so if the piece of DNA has a T, what do you put into the messenger RNA?
No, A.
A.
Wait, you tried.
I was not trying to trick you. All right, if the piece of DNA has an A, then the messenger RNA has a U. And if the piece of DNA has a C, the piece of messenger RNA is a G. Okay? And then it's a G, so what does the RNA have in it? C, C, U, okay, et cetera, right? And this is our 3' end, okay? In a question like this on an exam, we would never ask you to intron and exon and everything. You'll just assume that the piece of RNA is ready for translation, even though you know at a molecular level that's not true. All right? We read off these pieces of messenger RNAs in groups of 3. So everything's encoded in what we call a codon. So codons are 3 RNA bases, and they give you 1 amino acid. So we read in groups of 3. So the best thing to do, I think, is to just go ahead and underline in groups of 3. Okay? So functionally, we know that we start translation at the start codon. Everything that comes upstream, that is before, that's considered your 5' untranslated region. So do you translate the untranslated region? No. Okay, so we always start at the AUG. Now I've given you a codon chart, and I've shown you how to read it. The codon chart, you find your first letter, you find your second letter across the top, and then you find your third letter. AUG is methionine. Methionine is our start codon always. The next amino acid is CCU. All right, so first letter C, second letter C, third letter U. I can't read through the paper anymore. All right, so what did you get? CCU. I got proline as well. The last one is GAU. What did you get? GAU. Okay, and so on and so forth until you get to one of our 3 stop codons. Okay? What? Yes. Hang on. No. Do not memorize the codon chart, for the love of God. If you need it, it will be given to you. Okay? Yeah, go ahead. I'll give you both, probably. Oh, yeah. So, guys, on the back, what I need you to do tonight before we come back into class on Friday is work on the what will it look like. On the back, I've given you a circular codon chart. They work the same way. I'll give you a square and a circle. Circles, you work your way from the inside out. Some students like the circles better. Some students like the squares. I'll give you both, okay? Yes, guys, work on this. Make sure you can do this before you come back to class on Friday, okay?